Dear students, Assalamu alaikum. Today I shall take my second class about cell division. I expect you remember my previous class where I have discussed the significance of different kinds of cell divisions among various types of living organisms. I have noted a link here in Khan Academy for your clear conception about this chapter. Please read the article later and watch the video clip. This diagram shows the cell cycle. We are studying cell division, and in our syllabus there is mitosis and meiosis is there. To understand mitosis, we have to understand the cell cycle. It starts with G1. G means gap or growth, both are correct. So first gap in G1, the cellular contents excluding the chromosomes are duplicated. S comes from synthesis of DNA. So all the DNA material, they will be duplicated in this phase of cell cycle. In G2, the cell double checks the duplicated chromosomes for error and make repairs if necessary. M phase contains the mitosis and cytokinesis. So we can see a diploid cell here containing, for example, here it is a diagram, simple diagram, and shows only 2 chromosomes, one from each parent, but actually, for example, in Homo sapiens there are 46 chromosomes, 23 from either parent. So this is the S phase where the DNA is actually replicated, and when I shall discuss the mitosis, I shall show that there are chromatids and other terms are there for our understanding. This is overview of mitosis. Interphase, in our previous slide we have seen the G1, S and G2. Actually these 3 in total is called interphase. So cells don't rest during interphase, they are very much busy. Cells rest only when they are in G0 or G0 phase, then that is their resting phase. But G1, S or G2, that is interphase, that means this is between one mitosis and another mitosis. So it is active stage of cell, not resting. So after interphase comes prophase. After prophase, before metaphase, there is prometaphase. After metaphase, anaphase is there, and the last step of mitosis is telophase. This is a diagram, and this diagram is better, more clear. This is the interphase, and we can easily see that the strand is more dark because they are condensed and they are short and thick, so they are easily seen during prophase or metaphase or later. During metaphase, the chromosomes, or here chromatids, they assemble in equatorial plane, middle of the cell. And during anaphase, the chromatids, they travel towards the end of the cells, both ends. So karyokinesis means division of nucleus, and cytokinesis means apart from nucleus, the cytoplasm and other organelles, they will be divided. So some scientists say that cytokinesis is part of mitosis. Some say it is outside mitosis. But in our book, in textbook, it is inside the mitosis. So we shall study like that. Okay, here is a diagram showing chromosome, and we shall discuss the important terms to be used in this chapter, like chromatin, chromosome, chromatid, and centromere. These are all genetic materials. We know that chromosome is composed of double-stranded DNA double helix, we call it. Actually, it is composed of DNA double helix is composed of nucleotides, and in nucleotides there are nitrogenous bases. We know that adenine, thymine, guanine, and cytosine, and there are proteins, histone proteins and non-histone proteins as well. So chromatin is loose form of genetic material, normal state of DNA RNA. But chromosome is condensed form of chromatin. And chromatin becomes chromosomes in the prophase of mitosis. That is very important. They become condensed while prophase goes on. So the term chromatid we shall use. Suppose here that the chromatids are duplicated. We shall call it sister chromatid, and this small round thing in between the chromatids arms, that is called centromere. It is very much important for our mitosis. We often call this a sister chromatid. This is both the sister chromatids, they are actually identical. This and this side, and this side, they are same. So at some point during cell division, they will be separated. The centromere will be separated, the chromatids will be separated. Then we shall not call them chromatid, instead we shall call them as chromosome. Okay, prophase in detail. In our book, prophase has been explained from the perspective of plant cell and animal cell. So I shall explain both cells here. We know the plant cell, they are surrounded by cell wall and they have definite nucleus containing nucleolus, and chromosomes are not that much condensed, so they are chromatin, which I discussed in my previous slide. In animal cell, we see the difference. There is no cell wall. Instead, there is cell membrane. Nucleus is there. Nucleolus is there. Chromatin is there. We see extra centrosome, which is not present in plant cell. Each centrosome contains 2 centriole, and before cell division, this centrosome is duplicated. That means when the cell is dividing, instead of 1 centrosome, there will be 2 centrosome at both ends. I am sorry to mention, students, there is a small mistake in NCTV book here in this labeling. Actually, nucleus doesn't contain the cytoplasm or centrosome. So the correct labeling will be like this: nucleolus, chromatin, and nuclear membrane constitute nucleus. So centrosome is separate, not inside the nucleus, and cytoplasm is obviously outside the nucleus. So pro means before. So prophase is a part of cell cycle which happens at the beginning of cell cycle. So we see that condensation of chromosome is here. This is the main feature of prophase. Not only that, it is so much condensed that chromosomes are visible by compound microscope. Nucleus becomes little bit larger. Okay, now prometaphase, which is before metaphase. So we can see that mitotic spindle started to form. This is again little bit different between plant cell and animal cell. In animal cell, we know that centrosome is there, and one centrosome contains two small organelle, which is called centriole. So this is one centrosome containing two centriole, and this is another centrosome containing two centrioles. They play major role in the production of spindle fibers. Fibers are microtubules which can maintain the shape of the cell, and they help in the pulling and pushing of chromosomes, especially through their centromeres. Plant cells, they don't have centrosome or centriole, but spindle apparatus is there which form from fiber protein. So in prometaphase, the nucleolus and the nuclear membrane start to disappear. We see another term here, kinetochore. Kinetochore is another important component. They are kind of microtubule. They attach the centromere of the chromosome to fibers, spindle fibers. Spindle fibers are also called traction fibers or chromosomal fibers. Metaphase. We can see the plant cell and animal cell side by side. Well, equatorial region or metaphase plate is at the middle of the cell. This is imaginary area or imaginary line like equator on the earth. So it is in the middle of the cell, and during metaphase we see that all the centromeres are aligned on the equatorial region. So they are here equally distant. The distance between the polar region where in our previous picture we have seen the centriole, from this polar region where centriole is there, from both the centrioles the distance is equal. How? Because they are pulling and pushing, the total force is same. That's why they are in the middle. Like in Bengali, if we do dori tanatani, so if both parties have the same amount of energy, then the rope is fixed. That kind of thing. During metaphase, they are pulled through these traction fibers, and kinetochores play a major role as well. We can see the astral ray here. Astral rays are developed from centriole as well. They are microtubules and they play important role in this total development here. So we can see in plant cell, although there is no aster ray or centrosome or centriole, but they manage somehow, they have spindle fiber as well, which is not originated from centriole. Okay, anaphase, it is after the metaphase, and we can see that the centromeres, they are traveling, they are separated now. So each sister chromatid and the divided centromere, they are traveling towards one pole. So they were before in metaphase, they were jointly present. They were called sister chromatid, but now we shall call them daughter chromosome or sister chromosome. They are traveling towards centriole. Sorry, they are traveling towards the pole, centrosome there. Okay, so this travel again is controlled by kinetochore microtubules. One important point here I would like to mention, that these chromosomes look like, during this travel, they look like V, L, J and I shaped, and they are called metacentric, submetacentric, acrocentric and telocentric respectively. Let us see another picture. So these names come from the position of centromere here. So it is almost at the middle, so both arms are equal, so it is V shaped. So this is called metacentric, almost metacentric. This is L shaped, and this is J shaped. So acrocentric means centromere is almost at the tip, but in telocentric, the centromere is at the tip, not almost but at the tip. This is telocentric. But we have to remember, telocentric chromosome is not available for Homo sapiens. So that is the anaphase. Now we shall explain telophase. We have seen in our previous picture that the daughter chromosomes are traveling towards the pole. So the chromosome number is not changed. It remains unchanged because they were duplicated and now they are divided into half. So actually 2 by 2. So number is same. Now we see that the daughter chromosomes, they reach the pole, both pole. So for plant cell and animal cell, actually telophase, we can say it is a reversal of prophase. So in prophase we have seen that nucleolus and nuclear membrane, both of them degenerated. But now we shall see that they will regenerate, and we shall see that the spindle apparatus, they will be disintegrated and gradually disappear. Another thing we shall see that the chromosomes will be decondensed. So it was thick. Up to anaphase it was thick, but now it will be decondensed, thin again. This will be elongated back into chromatin and form a mesh called nuclear reticulum. And nuclear membrane will be developed surrounding this nuclear reticulum. So after the reappearance of nucleolus and nuclear membrane, I would say that the nucleus 2 inside the cell, there is 2 equal nuclei, and up to this is karyokinesis. Now the question of cytokinesis. Cell will have to be divided. So this is little bit different for plant cell and animal cell. In plant cells, inside almost at the middle, small parts of endoplasmic reticulum, ER, endoplasmic reticulum, will be aggregated in equatorial plane and collectively start the cell plate. So sorry, I'm showing the wrong cell here, sorry. Here in the plant cell, they will create a cell plate. How? After the aggregation of endoplasmic reticulum in the equatorial plane. And for animal cells, we see that contractile ring on plasma membrane is move inside and they pinch off the two nuclei. They equally divide this area in the middle, and this is called cytokinesis. And almost simultaneously, we shall see that there's other cytoplasmic organelles like mitochondrion, or if it is plant cell, then chloroplast, plastid, etc. They will be equally distributed. So this is all about mitosis division. In my next class, I shall teach you meiosis. Thank you very much.